Hello and welcome. This is Columbia Basin Research's fourth video in a multi-part series on using Program SURPH. Program SURPH is a program created by Columbia Basin Research at the University of Washington's School of Aquatic and Fisheries Sciences. Program SURPH, Survival Under Proportional Hazards, was developed to analyze data from release-recapture studies of animal populations, in particular to relate the survival estimates from release-recapture studies to individual base and group-based covariates. Today in this fourth video, we will focus on your results after loading in your data and selecting your models in the first three videos. Future videos in this series will talk about hypothesis testing and models across multiple datasets, as well as offering various hints and tips on getting the most out of the program. Alright, so let's fire up the program. As mentioned in the previous video, when there are pending models for which there are no matching results, the user can double-click on Estimate, which causes the selected optimizer to be used to estimate the parameter. Here, you can see an example of output from the Fletch optimizer. The output includes the name of the optimizer, the active model name, the beginning function value, and the ending function value. Once the optimization is complete, the resulting estimates are used as new seeds and the optimization is repeated. This is repeated until no further improvement in the resulting log likelihood is found, as defined by the ending criterion in the optimizer setup dialog that we covered in video 2. Once the parameters have been estimated successfully, the results appear under Available Results in the form of Active Dataset, Active Model. SURPH provides two reports under the View Results heading of the navigation panel, the Probability Report and the Parameter Report. There are also plots that provide visual representation of a group's covariates or individual's covariates effect on survival or detection. The Probability Report takes the parameter estimates from the active result and translates them into survival, detection, and final product estimates, along with their standard errors, using the link function defined in the model. If you look here, you can see a probability report for a study with 10 populations and 3 intervals. The report is structured like the CJS estimates report, with a detection and survival estimate for each population and interval. The model that was used for this example assumed unique survival probabilities for each interval and common survival probabilities across populations. The model for the detection probabilities was kept at the default interval population specific setting. Here we can click on the parameter report, which reports the actual parameters that were estimated prior to being translated into the probability estimates. Here you can see the parameter report that corresponds to the probability report previously shown. The survival parameters in the parameter report correspond to the interval effects model seen here. The following table explains the relationship of the survival parameter to the interval effects model. Similarly, this table explains how the detection parameters correspond to the fully parameterized model here. If we click here, you can see the associated individual covariate plot for the individual covariate length. The smooth red curve is the modeled survival as a function of the selected covariate value. The jagged black line is a non-parametric estimate of survival as a function of the covariate. If you click the histogram button here, you can also superimpose a histogram of the individual covariate data on the plot. The x-axis for the individual survival plot is the covariate value. The y-axis on the left is the survival estimate pertaining to both the modeled survival function and the non-parametric survival function. The y-axis on the right side is the frequency applied to the histogram. Note that the controls at the bottom of the individual survival plot allow the user to select the population and interval of interest. If there is more than one individual covariate in the data, the user can select the individual covariate of interest. The user can also selectively display or hide the non-parametric survival function or the histogram. Since the non-parametric survival function does not depend on any model assumptions, the degree to which the model survival function fits the non-parametric function can be used to measure the degree of how well the chosen model fits the data. For details of how both the parametric survival function and non-parametric function are calculated, see Appendix B.4.1 of the manual. If we click here, you can see the associated group survival plot, showing a group covariate survival plot for the group covariate distance. As with the individual survival plot, the red line shows the modeled survival estimate as a function of the group covariate. The points, indicated by a plus sign, are the CJS estimates for the selected intervals. In this example, there are 10 populations, hence there are 10 CJS estimates plotted, one for each population in interval 1. As with the non-parametric survival function on the individual survival plot, the CJS estimates do not rely on the covariate assumptions and can be used to judge how well the selected model fits the data. Note that this plot uses the default setting of interval specific. If the user selects population specific in the controls at the bottom of the plot, the population specific survival function will be plotted, and there will be one CJS estimate for each interval of the selected population. For details on calculating both the interval specific and population specific group covariate parametric survival functions, see Appendix B.4.2 in the manual. SURPH also provides diagnostic plots for assessing how well the current model fits the data. These are number 1, model estimates versus CGS estimates, number 2, residuals versus capture histories, and number 3, normal QQ plot. If we click here, we can see the model estimates versus CJS estimates. CJS estimates are on the x-axis and the model estimates on the y-axis for the currently selected results. The CJS estimates are the MLE estimates of the survival and capture probabilities and thus the best parameter estimates. If the currently selected model adequately fits the data, then the resulting estimates should be close to the CJS estimates and the points should be on or close to the identity line drawn on the plot. Large deviations from the identity line may indicate that the model does not adequately fit the data. As we can see here, you may click on any point and the information about that point will appear on the lower left of the plot. Clicking here yields the residuals versus capture histories plot. The x-axis is simply an index of the capture histories. The y-axis is the corresponding Anscombe residuals. The Anscombe residuals refer to the transformations of the residuals that, if this model fits the data, follow a standard normal distribution, meaning that the mean of the residuals will be 0, and approximately 95% of the residuals will fall in the interval between -2 and 2. As we can see here, red horizontal lines are drawn at -2 and 2, and about 95% of the residuals should fall between the lines. As with the model estimates for CJS estimates, the user can select a point to get information about the point displayed in the lower left. As another example, here we can see a contrasting example of the residuals versus capture histories plot. Notice that there are many residuals outside the -2 to 2 interval, and the residuals with values outside of the -4 to 4 interval. The probability of observing values this extreme with a standard normal distribution is about 1 in 30,000, indicating that the selected model here may not be adequate. Next, I can click on the normal QQ plot. The normal QQ plot is another tool to assess whether or not the residuals from the model are normally distributed, indicating adequate fit of the model to the data. The x-axis is the quantiles of a standard normal distribution, and the y-axis is the standardized residuals. If the residuals are normally distributed, the points should fall on or near the identity line drawn on the plot. The normal QQ plot shows the capture histories for individuals removed, censored, in red, and those not removed in blue. Details of the normal QQ plot can be found in Sokal and Rolfe, 1995. Advanced export functions under the Advanced Export Functions heading on the navigation panel provide the capability of exporting both the covariance matrix and the correlation matrix for the current model. The rows and columns are numbered from 0 to n-1, where n is the number of parameters in the current model. The parameter order matches the parameter report previously detailed. This has been lesson 4 on using Program SURPH, in which we covered estimating your results. Future videos in this series will look at hypothesis testing and models across multiple data sets, as well as offering various hints and tips on getting the most out of the program. Please note that if for any reason you're having trouble with Program SURPH and these videos are not sufficient to answering your questions, you can find the user manual at cbr.washington.edu/analysis/app/surf. The PDF manual is there, and you can either load that into a web browser or you can view it in Adobe. If you're using Program SURPH and have any questions or comments, on that same page you may give us feedback via our user satisfaction survey. We would greatly appreciate any feedback you have in helping make future versions of this program more useful to our users. We hope that this video helps you get the most out of Program SURPH. Again, if you have any questions or comments in regard to Program SURPH, please fill out our survey as previously detailed. You can also contact Columbia Basin Research at web@cbr.washington.edu. Best of luck on your analyses, and thank you.